Hi there. I got to go mushroom hunting with my granddaughter Maisie today, and here are the things that we found. I thought it might be fun for you to see if you can match things that look the same. And we can start to look at individual characteristics about fungi that make them so unique and so different and so challenging to learn. Here I've matched all the ones from my basket that I think share similarities. You can see it that they're different sizes and in different stages of development. These little buttons that don't have much shape are the rudimentary beginnings of this species. And the stalk expands first, and then the cap begins to open at the top and look like a little hat or umbrella. I'm showing you the stalks, which have these black scabers on them. They're actually raised bumps. And here we see that when we cut the base, we get a staining reaction, where the tissue, when oxidized or exposed to oxygen, actually turns color. Here we see a bluish, and also we just saw a reddish color. This is a really good look at the raised nature of these black scabers. We call it a scabrous stipe. The tips of the scabers are blackish because of the pigment melanin, which is dark. Now let's look closely at the cap. I think it's an orangish color. And notice there are small little patches of dense hairs that make it look textured. Here we see a big arc shape where some animal took a bite, perhaps a deer. Notice this one looks similar to the last one, but the colors are different. It has the dots on the stalk. And look at that yellow foot. We call this the chrome-footed bolete. Chrome means yellow. So let's look more closely at this stalk. And the dots on this stalk do stick up like the last one into raised dots, but these are colored pinkish. Isn't that beautiful? You notice that the sponge layer under the little cap is made up of lots of tiny tubes. But what my favorite thing is about this species is the color pink. When I'm walking in the woods and I see this pink mushroom on the ground, I just get so excited. Here's a third species that I've grouped together because they look alike. And you can see dots on this stalk too, I bet. We call these glandular dots. They don't stick up as much as the others, but they're sticky to touch. You can see a slug has been eating on the stalk of the one to the right. Typical slug marks. And it has the sponge under the cap. Whoops, slugs have been eating up here too. They're little scraping marks of their tongue called a radula. And the cap of this one is also sticky. Do you see that there's dirt and needles stuck to it? And it even is trying to stick to my finger. That's because it makes a layer of sticky gluten to protect the cap. I love to eat this one too, but you need to know your mushrooms to do that. Here I have just one of these, and it's kind of old. When it was young, the undercolor was bright yellow, covered over by these raspberry squamules. Those are the long triangle-shaped tufts of hyphal cells or hair-like cells. And under here, the sponge, you can actually see the holes, the tubes. The holes are much larger than the last three we looked at. This puts it in a different genus, the genus Willis. Let's take a look at my basket find again. We've picked out several, but do you see the yellow ones in the middle? I found lots and lots and lots of those. Let's take a closer look. You can see that they have the same basic shape as many of the ones we've looked at. A stalk that's rather even in size from top to bottom, a sponge under the cap or tubes, and a very bowl-shaped, upside-down bowl-shaped cap. So what is different about these? Well, the stalk we can see has yellowish coloring to it. And rather than the sticky dots or the scabers, this yellow color is made up from fibrils that run down the stalk that are yellow. The cap color is a lovely chestnut brown or an orangey brown color, and the cap is dry and not sticky. So you can see on the table that I've sorted them into specimens that look alike. And here we have our first one again with the orangish cap and the black scabers on the stalk. And these two were growing fused together at the base. What I'd like to show you now is what happens when you cut into mushrooms or look at other animals that have taken a bite. As we saw before, this one stains. And so as I scrape the tissue away at the bottom of the stipe, you're now beginning to see that it has turned from a pure white to a little bit of a tannish color as it reacts with oxygen from the air. The one on the left has the little incisor marks of a rodent that has scraped away and left it bluish. What do we see when we cut a mushroom open and look inside? You can see that there are brown tunnel-like marks in this cap that I've cut into. And up close, they leave long traces. These are the markings of fly larvae. And here we see two little teeth marked from the front two teeth of probably a little shrew, another rodent. This one on the right is missing a lot of its stipe. And this is characteristic of what slug damage looks like on fungi. Here we see again on the cap, tiny little marks of a radula, the scraping tongue of a slug, eating away, often in round circles. I hope you've enjoyed this journey through today's mushroom finds. There's always so much to look at.